Hello, I'm part of the PhantomG, the Photonic Hybrid and Nanomaterials group of the CISEC, Aveiro Institute of Materials in Portugal. The group research areas are integrated optics, luminescent solar concentrators, nanothermometry, solid-state lighting, single-layer and single-molecule magnets. Since 2015, we have been using the CytoViva system that combines fluorescence microscopy, 3D enhanced dark-field imaging, and hyperspectral microscopy. We are using this system essentially in two research lines. The first consists in the study of the spatial emission homogeneity of nano and microparticles or thin films in 2 and 3D spatial dimensions by combining optical and hyperspectral imaging. By combining spectral resolution with spatial resolution up to about 100 nanometers, we can create hyperspectral maps to study the effect of the agglomeration of the particles on the emission properties. The other research line in our group is the study of the incorporation of luminescent nanoparticles in cells. CytoViva system enables us to locate the particles within the cells, and using hyperspectral mapping combined with 2D and 3D imaging, we are able to study these luminescent nanoparticles as ethers and intercellular thermometers in order to build up temperature maps of the cells.